this is about where we're at so far. So we've had most of our lab reports have come in, and we've had 2 midterms, and so we're a little bit more than half in terms of what's left over for the grade. So coming up soon is short report number 8, 9. So I think it's possible on the lab schedule I had it due this week, but there are no classes this week. So that's good news for you. It means you get an extra week, so you can hand it in next week on Thursday. If you hand it in early, that's fine as well. Not a problem with that. And then the following week, we have, where's that the following week? I'm just trying to think. Yeah, the following week is the lab exam. I got my dates correct. Hopefully I got those dates correct. And I'm going to just talk about the lab exam for a minute now. I haven't actually decided what the format is going to be. Usually for the lab exam, I have a combination of kind of written and practical activities. So for example, people would do a microscope test where they have to do a gram stain, focus something under a microscope. I have a spectrophotometry test. I'm just trying to think what they were doing, where you do some pipetting and you prepare some dilution samples and all that. So the lab exam is going to look at your labs, it's going to be pretty heavily focused on the data and the data analysis. So if you think about some of the activities that we did, we learned how to measure a specimen when it's viewed under a microscope, how big it is. We also talked about the magnification of a drawing. We produced several graphs. So be prepared to either produce a graph or analyze a graph that's based on the data that's done in the course. And there will be a little bit of theory in there. So I might ask you some theory behind, for example, explain the principles of gel electrophoresis or something like that. So like I said, I will talk about the format of this once I've had a chance to think about it. So that will probably be, I'll talk about it a little bit next week and give everyone a note on what to expect. And so hopefully, if you're looking to pull your grade up, the lab exam is a good place to get a good mark. And then we have the final exam. So again, I'll talk about the final exam as time gets closer. I know one of the big questions people have is does it cover the whole course? And yes, it kind of does. It does focus mostly on the last part of the course though. So that's topics, what is that, 13 to 20? So that's about 2/3, 3/4 of the course is going to focus on that. And so what I do is basically, for the final exam, you can kind of think of it as a double midterm. It's 2 hours and worth a little bit more than twice the amount. And so for the final exam, I take, there's 20 topics. So I think I do 50 multiple choice. So I basically pick 2 multiple choice from every topic and a few extra from the end. Probably, 1 true or false from each topic. And then most of the written stuff and long answer and matching is going to be all from the later topics. So if you have any questions about that, let me know. You can see we do have a date for that, December 8th, which is pretty much exactly 1 month from today. Today is November 9th. So I know the semester has been flying by. And yeah, so a little bit of time to get prepared for that. And like I said, watch for lots of hints for the final exam. Okay, so I know the other thing everyone is thinking, people are thinking, okay, is this the way it's going to be like this? Hopefully not. This is just a little joke I found on the internet. Hopefully, like I said, in my class, I'm going to try to have no surprises for the final, so we should know exactly what to study for when we get there. Somebody is asking, will there be a mitosis, meiosis, long answer in the final? No, no, there will not be. There may be a couple of multiple choice kind of questions or something like that, but all the long answer kind of written or matching questions are all going to be from the last part of the course. A good question, thank you for that. Okay, so let's talk about DNA replication and repair. So last day we were talking about DNA replication and just finished off talking about DNA repair. And I know I have kind of a summary slide here. So if you take a look, this is my summary slide, and we drew a DNA replication fork. So we have 5 or 6 different enzymes or proteins. So we have the DNA polymerase. Oops, there we go. We'll go back. We have the DNA polymerase. We have the helicase. We have the single-stranded binding proteins. We have the topoisomerase. You can see it has another name there, which is gyrase, but I'm being consistent with the textbook. I'm just going to use topoisomerase. We also have the RNA primase, which is usually just called primase. And then we have the ligase, the DNA ligase. So I do have a video for you from the textbook publisher. So it's one of those BioFlix videos. It's going to review what all of these enzymes and proteins are doing at both the leading strand and the lagging strand. So let's take a look at that here in a moment. I was going to share this with you as well. I saw somebody on the internet has used their old Kinnex kits to make DNA. I thought that was kind of fun. I happen to have one of these sets at home. And so if you're looking for something to do while you're studying biology, and here you go, lots of fun. Okay, so here's the BioFlix video. I'll play this for you. Like I said, this is kind of summarizes what we were talking about. And then I have a few other things to say about DNA replication. So I'll press play for this.
Every cell in your body is produced by cell division. Before each cell divides, it must copy its genetic material in a process called DNA replication. Understanding of DNA replication comes largely from studies of E. coli, bacteria that are found by the billions in your large intestine. Let's take a look at how DNA replication occurs in an E. coli cell. As we zoom in, we see the DNA. At the origin of replication, the two strands of DNA separate, serving as templates for making new strands. The result is a replication bubble. The bubble grows in both directions, forming two replication forks. Let's zoom in on one of them. Many proteins work together at the replication fork. Only some are shown. Here, the DNA is unwound, and DNA polymerases, shown in orange, build new strands of DNA. Original parental DNA strands are shown in dark blue. Newly formed DNA strands are shown in light blue. Because strands in a DNA double helix run in opposite directions, the new strands must be made in different ways. One new strand, the leading strand, is built continuously. The other new strand, the lagging strand, is built in pieces. First, let's focus on the leading strand. DNA polymerase builds a new strand of DNA by adding DNA nucleotides one at a time. Each new nucleotide must pair up with its complementary nucleotide on the parental strand. Adding new nucleotides works the same way on both the leading and lagging strands. Each piece of the lagging strand begins with a short segment of RNA, shown in red. A clamp surrounds the RNA and attaches to DNA polymerase, which builds the rest of the new piece as DNA. When the piece is finished, it is released from DNA polymerase. How are pieces of the lagging strand joined together? A different DNA polymerase removes RNA and replaces it with DNA. However, it cannot finish connecting the pieces. An enzyme called DNA ligase joins the pieces together. Growth of the leading and lagging strands continues on both sides of the replication bubble until there are two identical DNA molecules. Although bacteria are very different from humans, the process of DNA replication in bacteria is similar to what happens in your own cells.
Okay, so DNA replication. A couple more things to talk about DNA replication. So one thing is what goes on when all that machinery gets to the end of the chromosome. So it turns out if you're a bacteria or somebody who has a circular chromosome, it's not really a big deal. The replication, it kind of just goes around, and then eventually when it gets to the end, the two ends are joined together by ligase. But this is not the same with eukaryotes who have linear chromosomes because we have an end, and so there's a problem there. So let me kind of show you what's going on at the end, and we'll zoom in and see what the issue is. So there's an end of a linear chromosome. You can see we have a leading strand and a lagging strand. And if we kind of zoom in here, I'm looking at the lagging strand, we have those RNA primers. And the RNA primers are going to be removed, and we're going to try to put DNA in there. So let's take a close look here at what goes on. So the next to last fragment, so this one here, is not a problem. We can use our DNA polymerase, replace the RNA with DNA, and fix the gap with the ligase. But here is the problem, is we have this last fragment, and you need this 3' end as part of the enzyme. There's no 3' end here for the polymerase to grab onto and kind of make new DNA. And so what happens is if you have this little gap here, and it just kind of breaks off. So what you have is the new strand is shorter, and then the strand after that is just a little bit shorter, and then the strand after that is a little bit shorter, and the strand after that is a little bit shorter. So this is really bad. We don't like it when our DNA degrades. And in fact, this means that most of the cells in your body are quite, honestly, they're dying. That's what's happening. Your cells are dying. If you look at a skin cell, it survives about 50 rounds of replication, and that's about all it can do, because eventually the chromosome is going to start degrading into the essential genes of the chromosome. And this is one reason why we only live to be a certain age, and you know, why we get a little less healthy as time goes on in terms of the integrity of our cells. And it's something that we don't know a lot about this. It turns out some people seem to have a better ability to prevent this degradation, and apparently exercise and good diet helps, but I don't know how good those studies are. We do have one way to prevent this, and one way to prevent this is to add something called telomeres. So I want to talk about telomeres for a moment. This is just showing the end of the chromosome basically shortening, and the end of the chromosome is called a telomere, and eventually it's going to disappear and, like I said, get into those crucial genes. So what is a telomere? A telomere is a way we can kind of extend our chromosomes and try to, you know, at least slow down the degradation of these things. So they're actually repeats of DNA sequences, and they found out the chromosome ends, and they're made by an enzyme called telomerase. So there's a good name for an enzyme. It makes telomers, so it's called telomerase. So how does telomerase work? It turns out this enzyme has a little fragment of RNA in it. It uses that RNA as a template, and then it goes along and basically fills in the DNA. So it just makes some repeats at the end, and we're looking at you know, a few hundred of these, and we can at least slow down the degradation of our DNA. Now, this is mostly active in our gonadal tissue, which is a good thing because we don't want our offspring to have degraded, shortened chromosomes. We want our offspring to have good, healthy chromosomes. And most of our other cells, it's really only active in very low activities. Like I said, apparently exercise actually helps with this to activate the telomerases a little bit more. So I think I have, yeah, this is just a continuation of that slide. You can see there's a telomerase, and it's extending the chromosome here and there. There's a little video there you can check out on YouTube if you're interested. I encourage you to go check it out. It's 1 minute, and kind of shows the process there. So a couple other things about this. Just let me go back. Never mind, that was on that slide there. Right here, I'm looking at this here. So this is telomerase RNA. So why am I writing this down? I'm telling you one of my first promises about the final exam. We're going to talk about 6 types of RNA over the next couple of weeks. And there's number 1. And so on my final exam, I haven't decided the question yet, but I'll tell you what I did last year. I will have a question, and it will be worth 6 marks of your final exam. So, and it's going to have the 6 types of RNA. So I haven't decided what type of question it's going to be yet. It could be a matching question, it could be a fill in the blank. So last year was a fill in the blank where I just had the 6 types of RNA, and I had either the name of the RNA or I had the description. And people just had to fill in what was missing. And for some reason, not everyone got 100% on that. Okay, so I'm going to be reminding you about the 6 types of RNA question again and again and again, and I will even have a table in one of my future notes that will show all 6 types and all 6 definitions. So make sure you learn all 6 types of RNA. So there's number 1, telomerase RNA. So a good one to know. So there's the telomeres. I'm not sure how they're stained, but they're shown there at the ends of the chromosome. Like I said, telomerase is highly active in the gonadal tissue to protect our gonadal cells for our offspring. There's a lot of research on these things, people trying to figure out if there's ways to extend them, and some people believe that if we could extend our telomeres, then maybe we could slow down aging or something like that. And if you Google it on the internet, you'll see there's lots of interesting theories about it. I'm not sure how many of those theories are well founded in science, but it may be possible. I don't know a ton about telomeres, but certainly there's definitely some interesting hypotheses out there about them and some interesting research. Okay, so I wanted to kind of finish today talking about DNA manipulation. Now, this is kind of a preamble to topic 19. So topic 19, we're going to be talking about biotechnology and forensics. But the reason why I'm pulling this up now is because a lot of these tools we've seen already, and a lot of these tools are using some of the same enzymes that are involved in DNA replication and repair. So it's kind of worth mentioning them here today. So 3 of the big tools that have been thrown out there, and maybe I should have added gel electrophoresis to this list, that are essential for modern molecular biology labs are using enzymes for cutting and pasting DNA, copying DNA, and proofreading DNA. So I kind of want to just talk about these briefly here for a minute, and we'll come back to them in topic 19. So first thing is cutting and pasting DNA. I kind of briefly talked about endonucleases being part of DNA repair, and some of these endonucleases are called restriction endonucleases. I'm not sure why they have that particular name, but probably because they're restricted to certain sequences. So you can see here's a whole bunch of these, and they're recognizing these sequences. So you can see we have this one here called ALU1, and it recognizes AGCT. So the interesting thing about this is that AGCT, we read that in the 5' to 3' direction. If I read the other sequence in the 5' to 3' direction, it also reads AGCT. So that kind of thing is usually called a palindrome. Palindrome. Right, so there's some interesting palindromes out there. For example, you could, if your name's Bob, or if you had a race car, right, it reads the same forward as backwards. And so this is kind of interesting, these sequences. And they can be recognized. And so now what you have, if you have these enzymes, I was just going to point this one out here, EcoR1, right? So that's from E. coli. They're often given code names. So there's hundreds of these sequences that have been recognized, and hundreds of these enzymes. And so these are kind of like precision scissors, right? So we can go in and we can cut very precise sequences of DNA. So this helps us to manipulate it, right? To edit DNA in a sense. And so this is showing if we use restriction endonucleases along with ligases, remember ligases stick covalent bonds together, we can make something called recombinant DNA. So you can see in this case here, we have some DNA, we're cutting it with EcoR1, and we have some other DNA, that stuff there on the, that pink DNA there on the right. You can cut them, put them in a test tube. They're going to complementary hydrogen bond with one another, and we're going to kind of seal the gap with DNA ligase. I think I have another slide here showing that kind of thing. Maybe that's later on. So why would we do this? Well, this is very useful for biotechnology. One of the oldest examples, and I think I mentioned before, is back in the 1980s when people took the gene for human insulin and they put it into an E. coli plasmid. And this is the type of technology that they used. So now you have a human gene in an E. coli plasmid, and now E. coli can be used to produce human insulin. So it's a really, really amazing technology. And this kind of thing, you know, I could hire a summer student to do this and probably do this for any gene, and we could get it done in, you know, in theory, a couple of weeks. It usually takes a little bit longer than that, but really amazing that we can do this. A lot of these plasmids, so this is the Biology 107 plasmid, and you can see it has the kanamycin resistance, so we talked about that in the lab. It's got the origin replication we talked about last day, and then over here, all of these little symbols and these NRI1 and Cas1 and all that, those are all restriction sites. So that's where we can cut the plasmid and open it up and allow, put in a new gene for it. So pretty cool stuff. So what else is another useful technique? Something called PCR. So PCR stands for polymerase. And polymerase, this really means DNA polymerase, but polymerase chain reaction. So this is a very, very powerful technique for amplifying tiny amounts of DNA. So you've probably seen crime scene TV shows or movies, and they find, you know, at the crime scene, they find a single hair, or they find a little bit of blood or semen or something like that. And we can analyze the DNA from tiny, tiny, tiny amounts. This is the guy who invented PCR. I haven't read his book yet. I've been meaning to look it up. Apparently it's interesting. He talks apparently a lot about surfing and the drugs he took too. He's apparently an interesting guy. But not condoning surfing or drugs. But you know, if you do read the book, let me know. I'm curious what it's like. Very important technique. Like I said, you know, this is one that you've probably heard about from the movies, forensic and paternity testing, and we're going to talk about that in topic 19. Pretty much anybody who does any kind of genetics or biochemistry kind of research nowadays gets involved with doing a little bit of PCR. And so you can see I have there listed genetics research. There's also some interesting research done on what's called paleobiology. So people looking at DNA of extinct organisms such as mammoths. And if you had a COVID-19 test, then they've used PCR to look for the viral RNA. And there's a whole bunch of viruses and bacteria and otherwise that's the test they use. So why do we use a DNA test for COVID-19? Not necessarily that it's fast, but it's very, very, very sensitive. And so it's a good test for that kind of thing. So what does PCR, how does it work? So PCR is kind of like, we just talked about DNA replication, and remember we had 6 things we had to worry about, right? We've got a polymerase, we have a primase, we have a helicase, we have a single-stranded binding protein, we have a topoisomerase, and we have a ligase. Okay, so that's kind of exhausting to think about all those things. And so this guy, Kerry Mullis, he said, well, what if we just put some of those things in a test tube? Is there some way we could make this whole process happen? So you need the DNA polymerase, right? That's the thing that is copying the DNA. So that is essential for sure. And we need the substrate, which is the nucleotides, right? And we need the template DNA. So hopefully those things are really, really obvious. We need the enzyme and we need its substrates. So, okay, throw them in a tube. And now we have to deal with all those other things, right? We have to separate the DNA. We have to hold it single-stranded and those kind of things. And DNA polymerase, remember, needs to be primed. It needs an RNA primer. So what we can do nowadays is we can talk to chemists and we can say, hey, can you make me a primer for this gene? So maybe we're looking for the coronavirus, a gene for the coronavirus, because we're doing a diagnostic technique. So there are companies that you can like, you know, order these things online and they come in the mail. and it's something like 50 cents a base pair. So super cheap. You know, spending about 15 bucks on some primers, and then you throw this all in a test tube. Now, the biggest innovation here is figuring out how this whole thing works, right? So you can see what he did 3 steps. So a denaturation step. So rather than using helicase, we heat the DNA up to a really high temperature, so near 100 degrees, 96, and that melts the DNA. The hydrogen bonds separate. Then we let it cool down, and this DNA polymerase can bind and do its stuff. So notice I have here, it's a heat-stable DNA polymerase, so we get it from some sort of a thermophilic organism, and it does its stuff. It copies the DNA. And then you do this again and again and again. And if you do 20 to 30 cycles, you're looking at millions of copies of DNA. So go on your calculator and just go 2 times 2 times 2 times 2 and do that 20 times and see how many copies of DNA you get. So I'll show you a little picture here of the whole process happening. There's the DNA. We heat it up. So they're using a different temperature, 94 degrees. There's a bunch of different protocols on this, by the way. That separates the 2 strands. And then the primers are thrown in. So like I said, these primers are made by a company that does the chemistry for you. And in fact, I think there's even machines that make primers nowadays. They're so commonly used. You throw in the enzyme binds to that 3' end. It does its thing. Where it sees an A, it puts a T. Where it sees a G, it puts a C, and so on, and goes basically to the end of the strand. And now we have 2 copies of DNA, right? So that's great. And then we do it again. You go from 2 to 4, then to 8, 16, 32, 64, and so on and so on and so on. And like I said, if you do that like 25 times, you're getting like 1 million copies. And so it's a very, very powerful technique used for all sorts of things. So we will come back and kind of review PCR and these concepts in topic 19. But like I said, they're worth introducing now because they're using most of the same enzymes that we used for DNA replication. So one other kind of technique worth mentioning is DNA sequencing. This also uses the DNA polymerase enzyme. It's basically a type of PCR, but in this kind of PCR, I'm not going to talk about the whole procedure here, but in this type of PCR, they use fluorescently labeled nucleotides. So you can see that A is green and C is blue and T is red. I don't know what color G is. There's no black fluorescence. But basically, the advantage of this is you can do the PCR, and there's a machine that will, in a sense, scan the DNA and figure out where all the nucleotides are and give you the sequence. So this is huge. This is important. This is how we discover new viruses, like we did in January when we discovered SARS-CoV-2. This is how we can proofread our sequences. This is how this is really a huge cornerstone of the modern genomics era. So many uses. And like I said, I don't want to get into the details of that. It would be an entire lecture to try to explain how this is done. Okay, so I think it looks like I'm at the end of this topic, and there's 10 minutes left. So I think what I'm going to do, rather than starting the next topic, is we'll break now. So I will not be seeing you on Wednesday or Friday unless you want an appointment. So hopefully everybody knows that Wednesday is a day off. It's Remembrance Day, and Thursday and Friday are the reading break. So there are no classes scheduled for that. So I will be working. If you do want to get a hold of me, I'll be working Thursday and Friday and open to appointments, but there will be no classes. So that's great. Hopefully it gets you a chance to catch up on things. I know I'm looking for a chance to get caught up on a few things, and a little bit of break, and then we'll go into the final stretch before the final exam. So have a great Monday, and I will see you, not this week, but I will see you next Monday. So take care.